molecular allergy diagnosis, I was asked essentially to give a very basic introduction for this first webinar. So I will talk about very basic principles and definitions of component-resolved diagnosis. Then I will go on with definition of allergen families and how this can help us to better understand what we frequently observe in our patients, co- and cross-sensitization, and how this can help us to disentangle this diagnostically. In the second part, which will be shorter, I will talk a little bit about methodology of IgE measurements. So essentially, allergy diagnosis is not too complicated. We start as basically in any field of medicine with a careful patient history to get an idea if what we observe in our patient is most likely due to allergy and which allergen sources are candidates of causing the sensitization and the symptoms in our patient. In the next step, we try to scrutinize our hypothesis essentially by IgE testing and by provocation tests, mostly skin prick testing. Well, since more than a century, we do this by using allergen extracts, but in the last few decades, more and more allergen molecules became available for these confirmatory tests. And based on the results, finally, we are able to give targeted advice to our patients if we can recommend immunotherapy or which allergen sources specifically the patient should avoid, or if we rather go for symptomatic medication. Well, one could ask the question, if allergen extracts work since a century, why just don't we continue using them? The question is, the answer is a little bit more complex. But first, to better understand what is behind extracts and molecules, I would like to talk a little bit about a few basic definitions. So what is an allergen source? This is quite simple. It can be any natural or man-made material like plants, food items, or animals which contain and release allergen molecules. And allergen molecules are in almost all cases, there are some exceptions, but almost all of them are proteins which can induce IgE sensitization in susceptible patients, which when the patient sees these molecules again can cause symptoms. So whenever we want to prepare an allergen extract, we collect specimens from such an allergen source, put the material into a buffer substance and treat it according to different protocols so that this tissue of the specimen falls apart and releases its molecules it is composed of. The majority of these compounds don't have anything to do with allergy. They are other constituents of the tissue, but some of these molecules are specifically involved in the pathophysiology of allergy. And those are the ones which we are interested in. It's the allergen molecules. In some publications, you just read allergen or also components, but essentially everything refers to allergen molecules. And this is what we use when we are doing molecular allergy diagnostics. Sorry. And likewise, you can read different designations for this concept. The original term was component-resolved diagnostics, CRD, and more recently, a new term was coined, precision allergy molecular diagnostic applications. But essentially, all of these mean the same. So coming back to the question, why do we need these allergen molecules? Why just don't you go on using allergen extracts? The answer is that even though they work to a certain extent, they convey some problems. And in the next few slides, I give you 4 different examples from 4 different allergen sources. In this first example, a colleague from Strasbourg, from France, Anne Cassé, she was analyzing commercial extracts from house dust mites. So this is number 2 to 9. This is 8 different commercial extracts to be used for skin testing and likewise for immunotherapy. Extract number 1 was made by the authors themselves. And here on this protein gel, what you see at the first glance is that these extracts are of considerable heterogeneity. While some seem quite nice and complete, like number 2, for example, and also number 7, others like number 6 or number 8, they appear almost empty. And if you look at greater detail at the allergen content of these different commercial products, you see that there's a considerable discrepancy in not only in concentrations of clinically relevant allergen molecules, but even worse, in some products, some allergens which are clinically relevant are even missing. And one prominent candidate, which was frequently absent in allergen extracts, is DERP23. Another problem which was found, for example, in dog extract, is that the extract can be contaminated with specimen from other sources. So if you think back to the previous slide, how an extract is made, it is based on specimens of an allergen source. So if you collect fur of a dog to prepare dog extract, it is quite apparent that this fur can be contaminated with other stuff, like house dust mites. And this has immediate consequences on clinical diagnosis, on test results. So these are results from skin prick testing of 8 different house dust mite allergic subjects tested with these 9 extracts shown in the previous slide. And if you look, for example, at subject number 8, shown on the left at the bottom, you see that there's a considerable discrepancy in results from skin prick testing. Here on the y-axis, you see the real area obtained in skin prick testing, and you see that this patient was negative with extract number 4, while he showed quite nice reactions with extracts number 1 and 2, for example. But this does not necessarily mean that extract number 4 is worse and does not work at all, because if you look at another patient here, top right, patient number 22, you will see that extract number 4 performed pretty well in this patient, while he was negative, false negative, apparently, with extract number 3. And the reason for this is that each patient has his particular sensitization profile, and it now depends on the composition of the extract. Does it match the patient's sensitization profile so that you get a positive result or not? Are allergen molecules the patient is sensitized to missing in this extract? The same is true for other respiratory allergen sources like Timothy grass pollen. Here on the bottom, you see the analysis of the composition of 4 different commercial extracts. And again, you see that this product here, number 4, for example, seems quite nice and complete, while extract number 3 seems to be less complete. And if you look at greater detail at the allergen concentrations of these 4 extracts, you see that there is a huge discrepancy. For example, if you look at major allergen FOP1 from Timothy grass, you see an almost 10-fold difference between extract number 4 and number 3. While if you look at FOP5, another highly relevant major allergen from Timothy grass, there's an even bigger difference between extract number 1 and 4, almost factor 20. And like in the example shown before from house dust mite, this has immediate consequences, because if you take, for example, 10 different grass pollen allergic subjects and do skin testing with these 4 different extracts, you will obtain different results depending on which allergen extract you use. If you look, for example, at patient number 2, he has the strongest reaction with extract number 1 and an almost 5-fold weaker reaction with extract number 2. By contrast, in patient number 4, the picture is just the opposite. He has strongest reaction with number 2 and weakest reaction with number 1. And patient number 5, the pattern is again different. He has strongest reaction with number 1 and weakest with number 4. And you can continue with other allergen sources. You will find the same discrepancy. Here, it's cockroach extracts. The Charts show concentrations of 3 major allergens from cockroach, allergy 1, 2, and 5 in different products. And you see at the first sight that there's again a huge difference. For example, here, allergy 5 is missing in some products. And with molds, it's even worse because molds are a bit different compared to other allergen sources, because mold spores and also the cells, they are extremely stable, and it's very difficult to open them when you try to prepare an extract. So they are in this respect even worse, and this leads to some extracts being almost devoid of any allergen content. Like in this study, some extracts were devoid of major allergens from Aspergillus or from Alternaria. So to overcome these problems, the concept of molecular allergy diagnosis was conceived. And as mentioned in my first slide, for this method, we use allergen molecules. In this respect, it is very important to differentiate between natural and recombinant allergens. And what is the difference? This is quite simple. A natural allergen molecule is purified from a natural allergen extract. It means that you, again, like when you prepare an extract, you take a specimen from an allergen source, make an extract, and from this extract, you prepare the desired allergen molecule. And whenever you see an N in minor letters ahead of the designation of the allergen molecule, like here from peanut, RH3, this tells you that this is a natural allergen molecule. By contrast, a recombinant allergen is based on isolation of the mRNA from the natural allergen source, encoding the desired allergen molecule. And after reverse transcription, you get the DNA encoding this protein. This you put into a vector, plasmid, for example, then you transfer it into cells of the in vitro expression system. And these cells, mostly used of bacteria, they kindly produce the allergen molecule which you are interested in. And then from the cell lysate, you can purify these molecules. And this you can identify by an R in minor letters ahead of the designation of the protein, like here, BETW1 from birch pollen. So essentially these two approaches exist, but what is the difference between them? If you look at natural allergens, it is apparent that this is the original allergen molecule which the patient encounters when he has contact to the allergen source. For some allergen molecules, isoforms exist. What are isoforms? These are different variants of essentially the same protein. They are mostly identical, but they have subtle differences. This becomes important if these differences represent epitopes. So this means that some isoforms could have epitopes which other isoforms don't have. So in such a case, it is crucial to have all isoforms which can bind IgE available for diagnosis. Some disadvantages or problems associated with natural allergens are rather technically in their nature, because if you want to purify a natural allergen molecule and this protein is present at very low concentration in the extract used for presentation, then the yield will be poor. Another problem which is frequently encountered is that extracts sometimes contain proteins which are similar in their physical and chemical properties. So if you attempt to purify one desired allergen molecule, it can be that it is very difficult or even impossible to separate it from similar proteins. So this means that your preparation in such a case is not entirely pure. And another protein which does not affect all natural allergens, but some, is that some allergens, natural allergens, they contain sugar moieties which can be by themselves epitopes, so-called CCDs, cross-reactive carbohydrate determinants. I will talk about this in greater detail in the next few slides. So if we look now at recombinant allergens, the biggest advantages are more technically, because once the expression system works, you can purify large amounts of these allergen molecules at constant quality. And as you use the DNA encoding the protein, you can be sure that the preparation is devoid of any contamination with other allergen molecules. And based on the expression system which is used, you can also be sure that recombinant allergens do not contain CCDs, which can blur your diagnostic results. Disadvantages or problems which need to be taken into account is that whenever you express an allergen molecule in an expression system, the purification contains only one isoform. So as mentioned before, if several isoforms of the same allergen happen to be important, you need to produce them all individually. And sometimes proteins can be really nasty things. It can be reluctant to be purified or expressed. In some cases, it is essentially not possible to produce an allergen recombinant protein. In such a case, you are limited to the natural allergen molecule. So talking a little bit about CCDs, what are these? Most proteins are glycoproteins. It means that in addition to the protein core, you have sugar side chains, sugar moieties. And considering the huge variety of monosaccharides which are available and the vast possibilities to link them one with each other, they can be linear, they can be ramified, etc. You have a huge variety of different sugars which can be attached to the protein. But luckily, only two patterns are common to CCDs, and this is shown schematically here. It is a monosaccharide named xylose and another one named fructose. And these two sugars, when attached in a particular way to this polysaccharide core, they can represent IgE epitopes. This is shown here on the bottom right. This here on top is a fructose-dependent IgE epitope, and here on the bottom, this is a xylose-dependent IgE epitope. So, as I said, they can induce IgE production, but the drawback is that these IgE specific to these sugar patterns are clinically completely irrelevant in 99.9% of cases. In very rare cases, these CCD-specific IgE can trigger symptoms, but in the vast majority, they are completely irrelevant. And why is that a problem? In this chart, in this slide, I show you 4 different scenarios which you may encounter in your daily practice. So here, this represents the solid phase of the test used for IgE measurement. These blue balls represent the proteins, the allergen molecules devoid of CCDs. And these golden structures here, they represent the CCDs. So if your patient now does not have neither IgE2 protein nor to CCDs, using such a test, you will obtain a negative result, which is essentially correct. It's true negative. If your patient happens to have IgE to the protein core of an allergen molecule in his serum, you will again receive a result which is correct. In this case, it is true positive. The problem starts here. In this example, the patient has IgE both to the allergen molecule, to the protein, but also to the sugar moieties in his serum. And in such a case, you will obtain a result which is essentially true positive, but the level of IgE which you are going to measure will be too high. The biggest problem is shown here on the right. In this example, the patient has IgE exclusively to the CCDs, and here you will get a false positive result in serological testing. False positive because these IgE will not cause any symptoms in the patient. But there are some strategies in order to overcome these problems, and I will talk about this a little bit later in my presentation. In the next few slides, we will briefly discuss the concept of allergen families. The background of allergen families is very simple. The definition is based on common amino acid sequences and, of course, based on these sequences on the three-dimensional structure of the proteins. And based on these two criteria, it was possible to define a huge number of different allergen families. 5 examples are shown here in this slide, and we will briefly discuss a few of them in greater detail. One very important allergen family is the PR10 proteins. PR stands for pathogenesis related, because they are thought to exert some defense mechanisms in the plant pollen. However, PR10 allergens can be found in pollen from many different trees. Here in Central and also in Northern Europe, birch tree is highly relevant as an allergen source. In North America, next to birch, also oak tree would be a representative where the pollen contain PR10 allergens. So these allergen molecules, PR10 allergens, they do not just convey cross-reactivity between pollen from different trees, but it can also lead to cross-reactivity with different plant food items from different fruits like apple or vegetables like carrot, but also from legumes like peanut or from soy. In most cases, symptoms of these food allergies are very mild. They are called oral allergy syndrome because they are restricted to the oral cavity, the lips, and the throat. In case of RH8 from peanut, it can be a bit more severe, but usually these symptoms are rather mild. Why is that? The background of this is that PR10 proteins are relatively unstable when you expose them either to heat, meaning when food is cooked, or to low pH, which is the case when you eat such a food item and it encounters the liquids in the in the stomach. So they are rapidly denatured and are not active anymore in terms of their potential to trigger allergy symptoms. Others, like PR10 from soy, glyme 4, they sometimes can cause severe symptoms. And the reason for this is thought to be mediated by higher stability of this protein or by other compounds contained in the soybean, which might protect glyme 4 when it goes through the gastrointestinal tract. This is the so-called matrix effect. Another family of allergens is the profilins. Profilins you will find in virtually any plant, and it is highly cross-reactive. This means that when the patient has IgE to one profilin, he most likely will react to all other profilins. And you find them again both in pollen, where they mediate mostly mild symptoms, if they mediate symptoms at all, but also in fruits and vegetables. And here I want to point out melon and banana, where the profilins can cause some nasty symptoms, but mostly also restricted to the oral cavity. Another very prominent example is the polcalcians. Polcalcians are also found in virtually any plant pollen. So please don't read the details of this table. This should just illustrate here on the right that there's a huge number of polcalcians which has been isolated and described and characterized. And here on the left, these arrows represent, each arrow represents a confirmed cross-reactivity between polcalcians from all these different species. So this should illustrate that polcalcians are also, like profilins, highly cross-reactive. But if you ask for the clinical reactivity, this is not always the case in each patient. So it means that some patients show clinical reactions mediated by sensitization to polcalcians, while others merely have any symptoms. A completely different family of proteins is one of the family of tropomyosins. This is very interesting, excuse me, because as they are muscle-associated proteins, they can be found in very different and diverse animals. You find them, for example, in house dust mites, in storage mites, but also in insects like in cockroach, shown here. But you also do find them in seafood, like in prawns, in lobster, but also in fish. So this means that the route of exposure can be the respiratory route, like in those examples, and also the gastrointestinal tract. And also the primary sensitizer can be seafood and then leading to a secondary respiratory reaction, or it can be that, for example, sensitization to tropomycins from house dust mite is at the beginning, and then the patient cross-reacts with tropomycins from other sources. And interestingly, if you prepare, for example, a dish which contains seafood, it can also be that the tropomycins are contained in the vapor, and the patient is exposed to tropomycins from seafood via the respiratory route, leading also to clinical reaction. In the future, I guess edible insects will become a more and more relevant source of muscle-associated proteins here in Europe, and I guess also in North America, because they become increasingly popular as a substitute for meat from mammals. And insects contain a large number of different allergen molecules, not only tropomycins, but also arginine kinase, sarcoplasmic associated calcium binding proteins, and myosin light chains, etc. So I think in the future we will have to consider these as possible sources of sensitizations in our patients. So all these ways to sort allergen molecules, to characterize them, can help us to better understand co and cross sensitization, and to use these principles to get to an answer what is going on in our patient. How does this work? So this is also quite simple. There are some so-called species-specific marker allergens and cross-reactive allergens. And if the patient now has IgE to such a species-specific marker allergen, this tells you that the patient is truly sensitized to that allergen source. On the other hand, IgE to such cross-reactive pan-allergens, they tell you that the patient is likely to show cross-reactivity to a larger number of different allergen sources. How can this help you? So if you now have a patient who complains about respiratory symptoms that start in springtime, which last over the summer until autumn, there are different plants and other allergen sources which could account for these symptoms. So in this chart, you see the pollen count in Central Europe. Here you have the 12 months of the year. In red, the chart shows you the average pollen count in the last few years, and in blue, the current pollen count. So in our patient, culprit allergen sources are birch, which are the pollen of which are in the air at the same time as the pollen from ash, which is allergologically distinct from birch. Another tree which can cause symptoms is plane tree. When you come to summer months, this grass which is in the air, but also spores from Alternaria, which has a very broad season, overlapping with the weeds which come late in summer, like mugwort or ragweed. So this should show you that based on patient's history and also based on extract-based tests, it is impossible to say against which allergen sources the patient is truly sensitized and what is due to cross-reactivity. And to further illustrate how you can solve such a case, I show you another very simple example. So in this publication, we had a young man who complained, like mentioned before, from respiratory symptoms, which started early in the year and which lasted until end of the summer. This patient was tested with extracts from 3 very relevant allergen sources here in Central Europe, which is birch, Timothy grass, and ragweed. And as you clearly see, is that this patient was positive to all 3 allergen extracts. And the question now is, do we have here a case of co-sensitization, meaning that the patient is truly sensitized to birch and to timothy grass and to ragweed. So we would rather recommend immunotherapy to all three allergen sources. Or do we have a case of cross-reactivity between these sources? And in that case, what is the primary sensitizer? This case was solved by using allergen molecules for skin prick testing. And the results you see here on top. This patient was positive to all three marker allergens from timothy grass, which is FOPI1, FOPI2, and FOPI5. So this indicates that this patient is truly sensitized to grass pollen. Looking at birch pollen, he was negative to the marker allergen for birch pollen, which is BETRI1. So in that case, we can rule out a genuine sensitization to birch pollen. So why did this patient show reactivity to the birch extract? It was not mediated by profilins, shown here, this was negative, but it was polcalcins. FOPI7 is the polcalcin of timothy grass, which is highly cross-reactive to BETRI4, the polcalcin from birch pollen. Regarding ragweed for this study, there were no GMP-produced allergen molecules available for skin testing, for in vivo testing. So this was resolved in vitro. And here again, the patient was negative to the marker allergen from ragweed, MA1, but he was positive to polcalcin from ragweed, MA9. So in this particular example, the diagnosis would have changed from allergy to birch plus timothy grass plus ragweed to genuine sensitization to only one of these three, to timothy grass, with cross-sensitization to birch and to ragweed. So also the recommendation would have changed from immunotherapy to all three sources to immunotherapy only to timothy grass. So this should illustrate how using allergen molecules can help you disentangle reactivity patterns, because each individual patient has his individual sensitization profile. So here I show you examples of 10 different grass pollen allergic subjects. So if each chart represents the sensitization pattern of one patient, here on the x-axis you see the different allergen molecules from timothy grass, FOPI1, FOPI2, FOPI4, and so on. Here the first bar represents the result obtained with the timothy grass pollen extract. And if you look at these patterns, you see that they are completely diverse. Many patients have actually IgE to the marker allergens from timothy grass, FOPI1, FOPI5. This patient, for example, has only IgE to FOPI5, but there are some patients, like number 5 here, who have IgE to FOPI4. And you see, as mentioned previously, this is N-FOPI4, natural FOPI4. This is a marker of reactivity to CCDs, because FOPI4 is clinically completely irrelevant, but it can give rise to a strong reaction in IgE measurement. You see, this patient had around 15 units of IgE when using an allergen extract, but in fact, this patient does not have any clinically relevant sensitization to timothy grass. So to put this on a more common ground, it is good to know that cross-reactivities occur between members of the same protein families. They do not cross the borders of different protein families. And in case of allergen families, which show very broad cross-reactivity, it is possible to define so-called surrogate marker for sensitization to the respective allergen family, like for example for profilins. This means that it is not necessary to test the patient's sample to all different profilins. It is enough to use one profilin, and if the patient is positive, he will be positive to most other profilins without the necessity to test the patient. This is true for some allergen families, but not for all of them, unfortunately. One very important example is the storage proteins from different nuts, legumes, and seeds like different globulins, 11S globulins, 7S globulins, and 2S albumins. Well, all of us know that these storage proteins can be really dangerous for the patient. They can cause very severe symptoms and even death of the patient. So these are highlighted here in red. Others shown here in orange, they can be very nasty. They can cause severe symptoms like the LTPs or particular PA10 proteins, but they can also cause only mild symptoms. And those highlighted in green, like Cori1 from hazelnut, they usually cause only mild symptoms. So, but if we focus now on the storage proteins here, unlike in other allergen families, it is not the case that all of them, all members of these protein families, show equally strong cross-reactivity with other members of the same family. This is shown here in green. The green allergens, they show limited cross-reactivity to members of the same family, which are close in terms of evolution. So evolutionary proximity defines in such a case if there's cross-reactivity or not. This sounds complicated, but luckily it is not so much. It is illustrated here in this tree chart. This tree chart illustrates the evolutionary proximity between the different species. And if we look at these two examples here on top, you see pistachio and cashew. This means these species are quite close to each other regarding their evolution. And this has a consequence because if a patient has IgE to 2S albumin from pistachio, he would cross-react with 2S albumin from cashew, but not with the corresponding 2S albumin from walnut or pecan. And it is exactly the same if the patient is sensitized to 2S albumin, for example, from walnut, he will cross-react with pecan, but not with cashew and pistachio. Another very relevant example is lipocalins, which are found in almost all furry animals. So you see that there's also quite different grades of cross-reactivity between the members of these families. But luckily, there's a scheme to diagnostically use this knowledge. By the way, these charts here are derived from the Molecular Allergology Users Guide of the IACI, which I highly recommend. I think it's open access. And this is a very nice resource to look up all these details on the different allergen families and marker allergens, etc. So if you happen to have a patient who is positive to cat extract, it can be that he's truly sensitized to cat or that he's cross-reacting based on sensitization to another animal, for example, dog. To solve such a case, you can use marker allergens. FeLV1 is the marker allergen from cat. If the patient is positive, this tells you that the patient is truly sensitized to cats. If he's negative, like here in this chart, this tells you that the patient is not really cat allergic, but his clinical reactivity and reactivity in other extract-based tests is based on sensitization to cross-reactive allergens, like here. And in such a case, you would need to do further tests using marker allergens from other sources like dog or horse. The same essentially is true if you have a patient positive to dog extract. Here you have marker allergens, can have 1 to 5, specific for dog, telling you if the patient is truly sensitized to dog or rather cross-reacting based on genuine sensitization to another allergen source. And the same is true for horse. For horse, reactivity to ACOC1 is not so unequivocally telling you that the patient is truly sensitized to horse. Here you have to consider reactivity to other allergen sources. So in some cases, it's a bit more tricky, but using these marker allergens finally will help you at the end to differentiate if your patient who is clinically and also in diagnostic tests reacting to several allergen sources, if you have a case of co-sensitization or rather of cross-sensitization. And ultimately, this could help you to increase the resolution of your diagnostic endeavor, to increase accuracy of your diagnosis. And this finally can help you to give more targeted recommendations on allergen avoidance or immunotherapy. And also, please bear in mind that extract-based tests always have the risk that you have false positives in case of reactivity to CCDs. So in the second part, which will be very short, I promise, I say a few words on methodology. First, if you do a IgE determination, you need to decide if you use a single-plex test. The most common example is the ImmunoCAP system, or if you go for a multiplex test. There are also several on the market, like ImmunoCAP ISAC, and also the Allergy Explorer, the Alex test from our company, Medex, and also the Euroline test, which is based on immunoblot technology. If you do a single-plex test, you need to first decide exactly which specificities you are interested in, and the result will provide you information which is limited on one allergen source or one allergen molecule per test. And it can be that if the patient happens to have a more complex clinical reactivity pattern, that you need to do several single-plex tests. On the other side, if you do a multiplex test, the results provide you with very comprehensive information on the patient's sensitization patterns. But to interpret such a test, you need to look into detail into all these links between the allergen molecules in terms of possible cross-reactivities and also the characters marker allergens. But luckily, there are some possibilities available to assist you with the interpretation. So now, as promised before, I said that there's a solution for this problem associated with CCD-specific IgE. It is a huge problem because 1/4 or 1/5 of patients has actually IgE to CCD in their serum. And as mentioned before, they are almost always clinically irrelevant, and they can lead to false positives in extract-based tests. But the solution is quite simple. Either you can use molecular allergens, more specifically recombinant allergens, which are devoid of any sugar moieties, or you can use a CCD blocker, which you add to the serum. Such a blocker, for example, is shown here. This is human serum albumin, and by biochemical methods, such CCD moieties have been linked to this human serum albumin. And if you now use this as a blocker, this works quite nicely. In this paper, serum samples were shown which reacted not to an allergen extract, but even worse, to the matrix of ImmunoCAP, which also contains carbohydrates. And in that case, these sera were non-specifically reacting to the matrix of ImmunoCAP, but pre-incubation with the CCD blocker eliminated all of these non-specific reactivities. And also in the Alex system, by default, we have a CCD blocker contained in the buffer, which is in the standard protocol used to dilute the serum. And also the efficiency of this blocking is quite nice. In this picture, you see the results of a serum without addition of the CCD blocker, and you see quite some reactivities. But if you pre-incubate the same serum with a CCD blocker, you see that almost all of these essentially non-specific reactivities could be eliminated. So this approach works quite nicely, to say. Briefly talking about the Alex microarray, you see that it's a large panel of allergen molecules. It's the largest panel on the market. Almost 180 different allergen molecules are contained. Half of them are derived from food allergen sources. Almost 1/4 is from pollen, and the rest is from molds, from mites and insects, animals and insect venoms. In addition, we have 117 different extracts on this test. Now, After having talked so much about problems associated with extracts, one could ask, why do you have extracts on this test? The answer is that not from each allergen source the molecular composition has been analyzed so far, and it is still better to have this allergen source represented as an extract on a test array than not to have this allergen source. And by using the CCD blocker, you can be sure that you won't have a CCD-related false positive result. So with this, I want to end and sum up very briefly. I hope I could explain why the use of allergen molecules ultimately is preferable compared to extracts whenever a sufficient a panel of allergen molecules is available. What is the difference of single and multiplex tests, and how multiplex tests can help you to solve more complex clinical reactivity patterns, and also how you can overcome the problem of CCD-specific IgE antibodies, which are frequently found in serum samples. So with this, I finish my presentation. I thank you for your attention, and I'm happy to take your questions. Thank you very much.
Thank you very much, Christian. That was a very interesting presentation. There are a few questions that have come in. You did mention toward the end of the talk that about 20 to 25% of people have anti-CCD IgE antibodies. How common do you think false positive allergy results are in the market due to CCDs?
Sorry, the question is how many false positives are expected?
Yes. Yes.
Well, I think at the end, as shown in one of my slides, it does not mean that the presence of CCDs leads to false positives in terms of a yes-no answer. Because if the patient is also truly sensitized to the respective allergen source, the level of IgE which you obtain at the end will be too high, but qualitatively, the result is not necessarily wrong. Yeah. The problem begins when the patient is not truly sensitized to this allergen source. And I'm not aware of any systematic investigation on a very large scale. But if you look at smaller cohorts, also restricted to particular allergen sources, you will find that this problem is very, very common, because please keep in mind that every fourth patient has IgE to these CCDs. Yeah, so it is really a huge problem, especially when you look at, for example, atopic dermatitis patients. I remember many colleagues sending in serum samples together with findings from extract-based multiplex tests, and these tests were positive for each individual items. And with such a result, you hardly can do anything, because you cannot tell the patient to avoid any possible food item and to do vaccination against all possible allergen source. Yeah, so you are somehow then lost with such a result. They are pointless at the end.
Okay. So another question. Do you think ultimately all extracts are going to be completely replaced by specific allergen molecules in the future?
Well, I think here we have to differentiate between extracts used for therapy and extracts used for diagnosis. Here in Europe, we have the problem that some years ago, the regulations of the European Union have been changed. This means that each allergen extract used for skin prick testing has to be registered anew, like a drug, for example. And this leads to the companies to limit and reduce their panel of allergen extracts offered. So at the end, I think we have the problem here that we will end up with a very limited number of allergen extracts. And it is still not clear what will be the solution to that. One option would be to make artificial virtual allergen extracts, which are composed of allergen molecules which are mixed together, like a better standardized allergen extracts, which might be used then for skin testing, but they have to be registered before. So I think there are several issues which need to be overcome. And for allergy diagnosis, I think at least for in vitro tests like ISAC or ALEX, I think in the future, step-by-step extracts will be replaced by allergen molecules. The better and the more we know about the molecular composition of the different allergen sources, also from sources from other parts of the world, that we step-by-step will replace extracts by the better defined and standardized allergen molecules.
Okay, and have you seen any impact on the adoption of molecular allergy diagnosis on the prescription of immunotherapy?
Yes, definitely. Already several years ago, there were some very nice papers from different countries where the authors had a look at changes in the behavior of physicians in terms of prescription of immunotherapy before or after using a molecular based diagnosis. And the result was that, as shown in the example, which I've given in my brief presentation, that before using allergen molecules and marker allergens, the physicians would have prescribed a considerably higher number of immunotherapy than what they would have done after having received the results of molecular-based diagnosis. The issue is that I think from the concept-wise, it is very clear that it does not make sense to prescribe immunotherapy to a patient against a source against which he is not truly sensitized. So I think this is pretty obvious and supported by loads of data. But from a regulatory point of view, to implement this into an official recommendation, I think the problem is that it needs to be shown that this changed behavior is superior to extract-based results. Yeah. The problem is that in such a study, you would have to prescribe immunotherapy to patients which, after knowing that this patient is not sensitized to such an allergen source, would not make sense, because treating a patient with an extract always bears the risk to induce new sensitizations, which not always are clinically irrelevant. Yeah. So the answer is that yes, there's a clear change in the outcome, in the conclusion the clinicians draw from these results. But I think from a regulatory point of view, we still need to prove that this technique, when it's about deciding for immunotherapy, is really superior.
Okay, and one last question that's come in. Are you seeing clinicians being open to adopting the molecular allergy testing in this panel format?
Sorry, if I see clinicians open to adopt it?
Yeah, the molecular allergy testing. Are they open to switching to this method?
Yes. I think I'm working in this since around 15 years, and I think at the beginning, many people were reluctant, especially when multiplex tests became available. They were really reluctant to use these tests. But I think if you look at this from a neutral point of view, it's like having a toolbox, and these multiplex tests are new tools in this toolbox. I always like to compare it with radiology. The old tests are like a normal chest X-ray, which in some cases do make sense, help you to come to a diagnosis. But these multiplex molecule-based tests are like the MRI of allergy diagnosis. And so after this long period of time, people became used to and familiar with this molecular allergy diagnosis approach, and also the literature was growing, and like this molecular allergology users guide, there are more and more resources available which help the colleagues to get familiar with these principles and with all these details which are needed to interpret tests. So step by step, more and more allergen molecules were used, and now for serological testing, they are routinely used here.
All right, thank you so much, Christian. Thank you to all of our attendees for joining us today. We know your time is important, and we appreciate you for sharing that with us, and hope you found the seminar to be worthwhile. We do have 3 short survey questions that will appear once the webinar closes. We certainly appreciate your responses. Thank you again, and please look for an invitation to the next webinar in our allergy series.